This is the last lecture, but I think is one of the most important because we'll give you the perspective on what kind of applications HPC is useful. It's not just like solving numerical systems, high performance computing and simulations, but also something that is going to change our workloads. And this has to actually encompass genomics, graph analytics. All of them are characterized by a lot of data movement, rate, and computational aspects. So it's my pleasure to introduce G.Guidi from Cornell University, and I have to keep the introduction short.
Yeah, no, please go.
So, Julia, please
Thank you so
go.
much. Okay, I don't think I'll be able to send you to lunch at 12:15, but I'll do my best to, stop in for like after 40 minutes so that we can all go for lunch. So today I want to talk about how we can use high performance computing and also a little bit of linear algebra. So I know that you had like a couple of hours of linear algebra lectures this morning. So I want to talk about how we can use that in actually biology. How many of you are somewhat familiar with biology? A little bit. Okay. So since I'm going to cut the presentation anyway, if you have any questions about the biology that you understand, please, feel free to ask as I go. I think it's much more, I rather talk about one things, but make sure that you understand that, put too much information. I'm just going to quickly say that, I want to thank all the people that have contributed to the work that I'm going to present today. This has been like a team effort. And in particular, it's been like a very interdisciplinary collaboration since the very beginning. And I think that that's really what makes this type of work extremely interesting for me. Like, it takes a long time usually to find a problem that is interesting from a scientific point of view and from an HPC point of view. But when you find that intersection, then the results can be like amazing and the work is really interesting. Okay, I wanted just to give you a little bit of perspective, since like many of you are at the beginning of their career or research career. I just wanted to give you a little bit of perspective of how my research career evolved. So I actually got my bachelor and master in biomedical engineering from the Politecnico di Milano here in Italy. And I took my first computer science course at the end of my bachelor, and I just fell in love with it. I never seen coding before that, and I just like love C and C++, which, maybe like that's a controversial opinion, but I really liked it. And so I really started to work with this professor, Marco Santabrogio. He's a computer architect. And I wanted to find something that was at the intersection of what I cared about, which was this kind of biomedical, bioengineering applications and computer architecture. And so we work on protein folding, which is, now it's very popular because of like AlphaFold and deep learning. But at the time, you were doing all of that through simulations. So it's like how the proteins goes into their like folds into their three-dimensional space, which is what gives the function of a protein. And we accelerated the simulation on FPGAs. Then I had the chance to go to the Berkeley lab for my master thesis. Berkeley lab is in California. And here I kind of moved away from FPGAs a little bit into focusing on shared memory parallelism and multi-core architecture, also focusing on DNA type of applications. And then I like California so much. I was actually planning to do my PhD in Milan, but then I decided that I wanted to go to the States. And so I got my PhD from UC Berkeley. And here I moved from simply shared memory parallelism to distributed memory parallelism. So this week you learn about OpenMP and you learn about MPI. And so that's kind of like also what I did in my career, but still focusing on biology applications. And as of January 23, I'm a faculty at Cornell University in New York State. And here I basically work on, all of the previous type of architecture and applications. So here it's like, and we add also like some specialized AI hardware, which, I was planning to talk about today, but I don't think I'll have the time. But if you, would like to talk about that, like I'm happy to do that offline and over lunch. So I wanted to use this kind of like architectural journey that I had for my career as an also an outline for the talk today. I wanted to give you first a little bit of background and context on the application. Again, so I saw that just some of you are somewhat familiar with the biology, so I just want to give you like a sense of the notion that you will need to understand the problem that we're going to talk about. And then I'll talk about how we can solve a specific problem that's called the novel genome assembly, and I'll give you more detail of what that means using distributed memory parallelism and linear algebra. So if you don't care about biology at all, that's fine, too bad. But I'll show you how you can use linear algebra, which you might like more than biology, to actually solve these real world problems. And then the second topic that I want to talk about is another common computational biology kernel on graph core IPUs, which is this AI architecture. But again, I'll probably not be able to talk about it. But if you're interested, I'm really happy to chat about that offline. Okay, so let's dive into the main topic, background. Okay, when we talk about genomics and biology and genomic data, there are actually a lot of different type of data that we can deal with. Like if you pick like four people from the biology department here, they will probably all work on different type of data and do different type of computation. So it's a very, very wide field. Think about computer science. Theory computer science is one field, but as you go dive deeper into that, there are like many different specializations. And so for most of my career, I've been focusing on DNA long read sequencing. Um, what is sequencing? Sequencing is this like physical and chemical process. It's like an actual machine. It looks like a big printer. It takes like a biological sample as input. It could be like saliva or blood or like something specific to the technology. And it basically runs this through this machine. And this machine, through again some physical and chemical process that changes depending on the actual company, return the string of DNA. So our DNA is composed of CTG and A's, and it returns the sequence of these characters. There's a biological sample, and then the output is this string. What makes this very complex is that because of like the limitation of the experimental limitations, the output is not actually your whole DNA. So it's not that like I input my blood sample and I get my whole string of DNA and that's what I'm gonna use and we're gonna do tests on that and that's great. It actually just can run short pieces of your DNA at any given time. So in practice, the output is a very large file with many sequences that are much shorter than your actual DNA. And so the process that we will talk about today is basically how do we take these sequences that we call reads and put them together to reconstruct your DNA. And that's what the genome assembly process is. And again, you might not care about the biology, but I'll show you how you can do this with like linear algebra. Does this make sense for everybody? Okay. Input blood, output a set of strings that are redundant because you want to make sure that you sequence every location of the DNA multiple times and contain errors because these technologies are not perfect. The first key observation when you work with this data is that the volume is growing significantly. And this is because the technology are getting the technologies that we use to sequence the DNA are getting much, much better. So this is the a very like a plot that everybody that works in this space will show you, but this is the years on the x-axis and the cost of sequencing one human genome on the y-axis. And you can see how in recent years the cost decreased significantly, much, much faster than Moore's law. And the idea here is that when you were, in the early 2000s, it was so expensive to sequence a single human genome that you didn't need a supercomputer to process it or like probably compute it. You just had so little data, and it was also pretty crappy that there was not much that you could do with it. But today we can do much, much more, and the outputs of the machine, which ended up being the input of our software, tends to be like several gigabytes to several terabytes. And so one key thing is like we have large volume of data and the cost of sequencing decreased a lot. The other good news is that the quality of this sequencing machine went up a lot. When I started to work on this data at the beginning of my PhD, the error rate was between 15% and 35%. And like 35%, it means that the whole, pretty much the whole string is wrong. Like it's a very high error rate to deal with. Today, most technology are around like 0.5% and 1%, which is significantly better, still creates non-trivial issues. And then we have some complexity that are intrinsic of the biology itself. The second key observation that I noticed while working on this type of data over the years is that there are some like sciences where performance is not a concern at all. Like people just wanted to get the results, they want to get the results correct, and that's it. Computational biology community didn't really care about performance, but they historically always rely on shared memory software. So they really never got into like GPUs or distributed memory, and that's something that is just starting more recently. We can talk about the fact that computational biology application have been like a computer science topic of interest from a product computing perspective for many years, but the biologists themselves didn't really took advantage of this until quite recently. And again, because they were like, share memory pluralism is kind of okay for the amount of data that we have, but today the scale is different. And the other issue is that multi-core architectures are not really scaling anymore. So we know that we are at the end of Moore's law, so we cannot really get more performance out of the multi-core architecture these days. So what we really need to do is like move toward distributed memory architecture and specialized hardware. Does this make sense for everybody? Any questions so far? Okay. And this is great. I mean, like I like distributed memory architecture, I like specialized hardware, so this is great news for me. Of course, you probably learned this week that programming with MPI, for example, is much harder than programming with OpenMP. Programming specialized hardware is harder than programming a CPUs, especially if you think about like novel hardware. I mean, like NVIDIA GPUs at this point have like a great software ecosystem, but that's not true for every like novel hardware. And so you know, with the scale, with the availability of like large scale and like massively parallelism of specialized hardware, comes also the cost of higher programming effort most of the time. The third key observation on this data, which I think might be the most interesting, is that genomic data are often very unstructured and noisy, which leads to very irregular computations. Like sciences have been like a topic of interest for like parallel computers for many years. Like think about simulations in physics or like mechanics. But even when we think about like sparsity, for example, sparse matrices, usually in science and engineering, these matrices do have like a nice structure that you can exploit to get better performance out of it. This is not true in genomics. Most of the applications, most of the data that you have are very noisy. Again, not just because the quality of the data is not always good, but because there are just a lot of things about biology that we don't know. And we do not know how to represent correctly, or we don't know how to filter out correctly. So this tends to be extremely messy and leading to regular computation, which from my point of view makes it like a very, very interesting applications. Sometimes I talk to people, they have like, scientific problems that are, extremely interesting, maybe from a scientific point of view, but from a parallel computing perspective are somewhat trivial. It's like embarrassing parallel, which I'm like, I would like to help, but from intellectually speaking, parallel computing perspective is not that interesting. Okay, so like as I anticipated, I think that what's really interesting about this problem is that you combine the need for large scale and performance with the regular computation. So the fact that you do not have like a clear structure that you can exploit easily for performance. And so the question here is like, okay, now we have this problem that is really challenging and it really needs high performance computing and really would benefit from parallel computing. How do we actually do that? How do we actually implement it on a supercomputer or on specialized hardware and actually get out performance out of it, right? Doesn't make sense to implement it on a supercomputer if it's, the speed up is 0 or like minus 15. We don't want to do that. And the answer to this question that we gave is genomics has a lot of graph structure. A lot of things in genomics are graphs. It could be relationship between sequences, relationship between genes, relationship between sequences of DNA and proteins. So a lot of things is like relationship between stuff, like biology unit. And this can be represented as graphs. And we know, or if you don't know, you will know now, graphs can be represented as sparse matrices. They're just can be represented as the same. Usually represent a graph as an adjacency matrix. So here, this is like a classical example. This is a graph that you connect how people cited each other, and then you can recreate the exact same structure using a sparse matrix. So where you have Bob and Carl on the rows, which are people that are citing somebody, and the columns here are people that are being cited. And then you can actually perform a breadth first search graph traversal to figure out who actually cited who by using sparse matrix vector multiply. And I will not go into detail of this example, but I'll show you how we can actually do genome assembly using this. Does this make sense for everybody? Okay. The caveat here is that Genomics usually does not deal with numerical things. So, um, this morning you learn about linear algebra, floating points, but genomics is not usually about floating points. There might be some application that are, but again, usually it's about relationship between pieces of sequences. So most likely your connection are strings or custom data structure that, means something for that application, but are not easily representable with a floating point, a Boolean, uh, or an integer. And so what I'm going to show you now is like an actual example of how we can take this linear algebra and use it for implementing like a computational biology applications. Any questions so far? Makes sense? Yeah. hmm Does it create problems? No, it doesn't create any problem. You can just you do not lose any information moving from the graph to the sparse matrix. Yeah. Yeah, but the sparsity is already in the graph. So like when I move, sorry, maybe I didn't understand your question, but like I do not modify, like, the sparsity is intrinsic of the data. So it's not that I have a dense type of connections and I sparsify into matrix. The connections are already sparse in the graph. Maybe it become more clear when I have the genomic example, but there's no loss of information in moving from the graph to the matrix. There might be an application where you want to sparsify more, but that's not what's happening here. Any other questions? Yeah. That's a very good question. I actually talked about that with a colleague of mine at Cornell, that he's scientific computing person more than numerical algebra, and he was like, well, it seems like a reordering problem. Why don't you do that? Uh, well, the issue is that this, like as I was mentioning, the data are extremely noisy and messy. So you would expect these matrices, and again, it will become more clear later. But if you think about the genome, right? This is your matrix. You would, and if you have row and columns that are sequences, you would expect a sort of like linear, like diagonal matrix, right? Because it's a linear sequence. But the reality is that the matrix that you start with has no zeros that are uniformly distributed. And it's really hard to just, it's like impossible to just find like an Hamiltonian pattern just because of the errors and the repetitions in the genome. But that's a very good question. Like it is a reordering problem, but you cannot really solve it. Like the data is so complex that you cannot really solve it just shuffling row and matrices around. At least with the quality of the data that we have right now. If biologists will learn more about how repetition works and the data will become perfect, maybe that will be possible. Okay. So we went a little bit ahead of ourselves, but the novel assembly, as I mentioned, we have this collection of reads sequences from the sequencer that are erroneous, and our job is to try to put them together in a linear sequence that is our DNA. And this would be simplish if we would have a reference genome and we would just map each read on the reference genome, but we want to do it de novo, which means we do not have any reference. And there are a couple of reasons why we want to do this. First, not many organisms have a reference. We might be interested in studying like animal and plant species that have not been heavily studied in the past year, and so we do not have a reference to start with. And second, there is a lot of, like, for example, for the human genome, we do have a reference that, contrary to what Wikipedia says, was not completed at all in 2003. In 2003, we had a sort of like sparse skeleton of a reference. It was actually completed, like, fully a couple of years ago, like 1 year and a half ago from a group at NIH. So now we do actually have a reference, but there is also a lot of research that shows that um, this reference can be biased. First, because the reference that we have is not an actual human being. It's like a combination of many of us. It's like if we take pieces of all our DNA and say, this is the reference, which, by itself doesn't seem like making super sense, but also is extremely biased towards certain populations. And we know that different populations carry different variants, or like, different like portion of variants. So like there is a lot of research showing that de novo is actually what, much more helpful if you can do that. So it's basically like building a puzzle. It's like somebody enters in the room right now, drops a huge puzzle on the floor, and leaves with the box. So we don't know if you're trying to build a cat, a dog. We just need to kind of like pair pieces together and try to reconstruct the whole thing. And one complexity that I want to mention is that the genomes do include a fair amount of repetitions. It's like this half of this puzzle is a blue sky. So when you have two blue pieces, it's going to be very hard for you to figure out where it actually goes. And the amount of repetition varies based on organisms. So bacteria tend to have close to zero repetition. Humans, somewhat in the middle, and plants, it's everything is repetitive. Like plants are extremely hard to deal with, and they tend to have much bigger genome than ours. Just so you know, onions have a bigger genome than ours. And here is some performance just to give you a motivation of runtime based on 48 core CPUs with multi-threading. And these are runtime from HiCanu and Canu, which are not the fastest, but the most popular that people do actually like and use. There is a newer version of HiCanu right now, but the runtime issues are still there. I do have some results at the end, also with the fastest version, but I think it was fair to compare to what people actually use and like. And here is the different scales. So the bacterium takes 1 or 2 hours, which probably you don't need a supercomputer to assemble a bacteria. But when you go into mammalian genome like humans, then it starts to be even in the order of days. And you want to think about the fact that like you don't usually do this once and then you're good for life. It's usually like usually you have many individuals to assemble. Maybe you want to assemble all of us, or you want to do it running multiple times to tune the parameters of the assembler. So it's not usually like once and done, but it's something that you do repeat multiple times. And the issue gets much bigger when you go into plants. Again, onion was assembled for the first time like 2, 3 years ago. This is a time estimate because I reached out to the authors multiple times to actually know the exact time, but I've been ghosted multiple times. So this is my time estimate, which I suspect is a lower, bound, just because again, as we go into plants, the amount of repetition increase, and that's extremely computationally intensive. So I think this is kind of a best case scenario type of estimation. And Paris japonica is a plant, and it's one of the biggest plant that we know. Like I think it might be the biggest genome that we know of. Okay, so this is just to give you a motivation of why we care about going fast. Now, what the overlap detection looks like. So there are like for the genome assembly, we have three big phases that basically every assembly uses. The first one is the overlap detection. So we have all this set of reads sequences, and we want to find like small pieces of these sequences that we will call seed or k-mers, as we will show, and find matches between them. Because the idea is that if you have one sequence and you know that they overlap here through this seed, then maybe you know that these two sequences overlap here with this, and then you can reconstruct by using this overlap as bridges. Does it make sense? Okay. And this is the step that we are going to focus on today. But once we have that, once we have all this pairwise information, we actually want to look at them from like kind of a global graph perspective. So now we have all these sequences that are all connected with each other through edges. And we want to try to simplify this graph as much as we can. So there will be some like transitive relationship between the sequences, right? If you have um something, let me think about uh like if you have a bunch of like connection here and here, you might be able to remove some of these without losing any information, but it will make your graph look simpler. And so that's what the layout phase does. And then in the consensus, you try to collapse that graph into a linear sequence. It's actually, usually not this clean, the process, but that's kind of the high level idea. And today I'm just going to talk about how we do overlap detection with matrices, but the reality is that we do everything with matrices. And so our software is called Alba. And so here, what we do is that we take the whole pipeline and we just represent it as sparse matrices and operations between sparse matrices. And again, and then I'll show you how that actually works when we do not deal with like floating points. And The good news about this part is that we get high scalability, high productivity, and a significant speed up. And why that's the case? Well, this is very irregular type of computation that it's really hard to parallelize in distributed memory because it's really hard to figure out what's the best way to do it. But once you move in the sparse linear algebra space, then we have, decades of research on how to make sparse matrix multiply or sparse matrix vector multiply faster and scalable. We have different algorithms that works better for different type, like number of processes and stuff like that. And we can exploit all of them. Like, is tomorrow one of you, comes out with the greatest, latest sparse multiplier algorithm, then we can just plug it in and use it. So we can continuously exploit what the community put out in terms of like linear algebra. Does this make sense? Questions? Yeah. No, these matrices are like 99% sparse, even more. So we use a sparse format. Here we use doubly compressed sparse columns. Yeah. But you could use other formats if you would use a library that use a different formats. But you want it definitely to be stored in sparse format because they're very, very sparse. Yeah, I wish. No, there's no pattern. We look at different genomes and we try to identify, but like all the data set that we've been looking at is pretty much like uniformly distributed sparse matrix. Again, there should be, like because we know that it's a linear sequence, there should be a pattern. But the data is messy and there are these like repetitions across the genome that just create edges everywhere. So there should be a structure, but it's nearly impossible to see just looking at the matrix. Very good questions. Okay. So here in this first slide, you saw like the K-mer counting. So I'm just going to briefly say what it is so that you can understand how we move from, how we got into the sparse matrices, but that's not the main topic here. So the first step that you want to do is to parse the DNA sequences that you have into these short substrings of fixed length K that we call K-mers. And K-mers is what we're going to use as seed to find pairwise matches between the sequences. Most implementation in distributed memory uses distributed hash tables. So what they do is that they divide the reads across across the processes. Each processor has some reads. It extracts the k-mers of its own reads. And then based on a hash function, they will be assigned to a specific processor. Like if you have like the k-mer ACG might appear on every processor, right? But you want only one processor to count that k-mer. And so k-mer ACG will always be hashed to one processor and sent to that processors. And so then, like, once the local processor has all the instances of that k-mer, it can do a local count and say, oh, I have 30 instances. It's basically like, this is like one student once told me, it's a glorified histogram. K-mer counting is a glorified histogram. So that's basically all it is. And all you need to know here is that usually it's done by using hash tables and distributed hash tables. We do have a recent work. This is like a little bit of advertisement that just got accepted at ICPP 24, where we implement this like k-mer counting by using sorting-based algorithms rather than hash tables, and we can actually achieve pretty good performance. But this again was just a little bit of advertisement. And so let's assume that we are still using the distributed hash table based implementation. Our output is this distributed hash table where we have the k-mers as keys, and the value is the frequency and from which sequences they came from. So if k-mer 1 appears in read 2, 3, and 4. So this is what we have. Then here is the last time that we are in the non-sparse matrix based environment, and then we move to sparse matrices, and we do not leave it until we have completed the computation. And we can move from hash table to sparse matrix pretty easily, similar from like how we move from a graph to a sparse matrix. So here we have a transpose that is our k-mers on the rows and our reads on the columns, and the non-zero is telling us that that k-mer exists in that read. And we have the same amount of information, right? Like if you take the number of non-zero per row, you will, sorry, per column, you will know how many k-mers that read has. Take the number of non-zero per column, you know how many reads that sorry, the frequency of that k-mer. So you do not lose any information here. Does it make sense? Okay, great. So the other things that changes here, which, in this presentation, I don't go too much into the actual, like, how we parallelize that, because I assume that, like, you will know about sparse matrix parallelization, but just to give you a sense, here you are in the 1D distribution case. So every processor has a subset of k-mers and all the information associated with that k-mer. While when we move to the sparse matrix, we actually distribute 2D. So every processor has a subset of k-mer and a subset of read. And we do this because we know from the theory that 2D sparse matrix multiply does scale better for large number of processors, because it scales with square root of p instead of p, where p is the number of processors. So that's what we want to do. But what's interesting to notice here is that while it's kind of like easy to see that you can divide a sparse matrix in 2D, it's much, much harder to think about how you can divide a hash table in 2D. Like, where do you decide the second dimension boundary? It's much, much trickier. And I'll come back to this in the results to make a point. Okay, so now we have our glorified histogram in sparse matrix form. So we have A transpose, which is the one that we just saw, where we have k-mers as rows and reads as columns, and then we have its transpose. The simple reason why I mentioned A transpose first is that because of how our code is written, it's faster to create A transpose and then transpose in A than vice versa. But that's just because of the sparse matrix format. Okay, so now we have these two matrices. What we want to do is like multiply them in order to obtain a final sequence by sequence matrix that will give us exactly this information. So how different sequences are connected. And that's just a graph. That's just a graph that says, read 1 is connected to read 3, and read 3 is connected to read 5, and so on and so forth. So if we look at read 4 and read 3, so we can see that they have, but I have the reflex, so I cannot see. So they have both k-mer 3 in common. They both have the same k-mer. And so if you do a matrix multiply, then you can put a non-zero in the R4, R3 cell. Does it make sense? Here you can assume you're doing like a Boolean or an integer matrix multiply. And if you do this for everybody else, you obtain this symmetric matrix. Of course, like the diagonal doesn't mean anything because yes, R1 doesn't lie with R1. But this is what you obtain. In practice, from now on in the computation, we just deal with the lower triangular matrix because everything else is it's not needed. Okay, so this is great. So now you do have a matrix that is read by read, and you know that they are connected, but based on that matrix right now, you have no idea how they are connected. You know that they are connected somehow, but you don't know if, and I think I have a picture, but you don't know if they are connected like this or like this, and so on. So you don't know the relative orientation, which is a very important information from the biology point of view. One could argue that, oh, you can recompute that information once you have the matrix, but why doing that? You had the information before. Why don't you just do the multiplication, include that information? Like, for example, we want to know how many k-mers, how many seeds they share, and exactly where, so that we can have that actually like relative orientation information. And again, as I was showing there, here is just some more example. If you just look at this matrix, you have no idea if they align like that, like this, like this, they could be anything, which is really the information that you want. And so the good news is that from linear algebra, we can actually exploit what's called semi-ring abstraction in order to change the matrix, sorry, the multiply and addition operation of the matrix multiply to do virtually whatever you want. If you want to be algebraically correct, you will have to ensure certain mathematical properties do apply. But if those mathematical properties do not matter for your application, that's fine too. You can still overload, addition, and multiply to do whatever you want. And this means that not only you can overload, addition, and multiply, but you can also have different types of matrices. So here you have A and A transpose, so they will have to have the same type, but C can be a different type. If you have A times B equals C, then A, B, and C can have different type, as long as the operators that you define, make it consistent. And so what we do is that we overload, add, and multiply, and basically the output type, so the type of the output matrix C, it's basically this structure, which is telling you it's an array of position of k-mers. So it's telling you how many k-mers they share and in which position on both reads. So now in C, we do not only store, yes, they share something, but we store that they share K2 and K3, and in this position, so we have the actual orientation of the reads. Does this make sense for everybody? I think simmering is very cool. Okay. And so if we assume that we went through the whole algorithm, so also the graph layout. So this is now, sorry, this is now a graph like this, but you do have this information. And so what you do next is like using also sparse matrix multiply and simmering abstraction, we clean up this graph by doing a transitive closure on it. And then using connected components and other type of algorithms on matrices, we extract the context, which is the linear finite sequence. Again, I'm going to skip on that so that we can all go to lunch. But here, if we implement all of this on a supercomputer using matrices, we move from days to under 20 minutes. And this is run on the Summit supercomputer, used to be the top 500 a few years ago. I think I don't know if it's in the top 10 now, maybe in the top 15. But and I think this was run on like 200 something nodes, and I need to double check it up. But so we can reduce the runtime significantly by using a lot of parallelism, and this represents a 150 more than 150x speed up compared to the original Canoe, and between 9 and 51x% of the fastest tool that we tested. And here is a comparison between ALBA, which is what I was talking about is matrices, and Dibella 1D. So Dibella 1D is also something that we implemented, and this is just overlay detection. So this two represents only overlay detection, which is the part that we talk about today. And Dibella 1D implements that using distributed hash tables. There are three things that I would like you to take away from this plot. First one, they both scale pretty well. Distributed hash table and sparse matrices with a good implementation, they both scale like 91%, more than 90% parallel efficiency. So that's pretty good. This is like over like 300 nodes. Then the other things is like ALBA is consistently 2 times faster, which is great. But I think that the most interesting piece of information here is that to develop the core overlay detection algorithm ALBA, it took more or less a month, Plus, then you can add some time for debugging, but like that was kind of the core time that it took us to develop the algorithm. The Bella 1D for the overlay detection was basically a PhD thesis. And not because like I'm any smarter than the person that did that. I have we worked together on that. It was just way, way harder because we started from the distributed hash table and we had to figure out all this irregularity and how to make it scale properly. While with matrices, that kind of came for free. It's like, oh, this is a matrix and we can just use Parsuma or whatever latest, greatest implementation that, somebody very smart came up with. And so that's really like, I think like a critical point here that if we can exploit linear algebra for this type of application, and there are many, I mean, this is an example, but we are working on many more, then we can exploit all this like research on dense and sparse linear algebra that a lot of smart people have been working on for decades. So that not only gives us performance, but give us time. It gives us productivity, which is critical because we are moving, to like many more, especially in this type of application, a lot more data. And it's kind of like, not realistic to think about that any bioinformatician on this planet will suddenly become a pro of MPI or CUDA or like whatnot. We really need to figure out how to make that easier to exploit supercomputing systems. Okay. And so here, to summarize, I show you how we can, really implement this large unstructured computation and achieving good performance using sparse matrices. And we can get speed up and scalability, which is great, which is kind of like the starting point. Otherwise, I will not be here talking about this. But I really want to stress out the productivity part because I think that that's really important. Again, my dream is that, one day computational biologists will do this without me doing this for them, but like they will just being able to exploit sparse and density algebra for their computation and get, quote quote free performance out of large scale system and specialized hardware. So here we talk mostly about, like this was just on CPUs, but we do have work on GPUs and specialized hardware, which again, in the interest of lunch, I will not talk about, but I'm really happy to chat about that offline. I guess the last things I'm gonna share is that we have like a new work that was just accepted at C24, which is on GPU-based pangenomic graph layout. So this is another graph-based computations, and pangenomic is an emerging field of genomics where basically people realize that looking just at the linear sequence of the genome of like my genome might not be that interesting. But if we take all our genomes and if we put them together in a graph form where we collapse the parts that are identical and then we create nodes and bubbles where we have variant, that might be very, very interesting. So that's what they do. And so, this is like an emerging area. So there is still a lot of developing that goes on the biology side, but it's extremely interesting. And they will always be happy of having more scale and performance. And so with this, I conclude the lecture. I cut it short, but again, if you have any questions on this or on the part that I didn't talk, but I will upload the slides with the whole presentation. So if you have questions on the second part, feel free to email me.
Thank you,
Thank you.
Julia.